Hello and welcome to this week's episode of Science Club. Last week's riddle was answered correctly, though not too elegantly, by Ignatius in grade 9. Congrats, Ignatius. The answer was no. It is impossible to place down 31 dominoes despite there being 62 squares available. Each domino covered exactly 2 squares, one white and one black. And in covering 2 opposing corners, you're taking away 2 squares of the same color, thus 2 dominoes. Before we get into the video, I want to remind all of you VC students to subscribe to our channel if you'd like to join Science Club. We would really appreciate it if you could also like and comment on this video, as making this club is really hard work. Today's riddle may seem challenging at first, but don't give up. The answer is much simpler than you might expect. The riddle goes as follows. A Buddhist monk is climbing a mountain to reach the top, where a great temple sits. The path is quite narrow and winds around the mountain. The monk starts climbing in the morning, stops occasionally and slows down to catch his breath, and manages to reach the mountaintop in the evening. After a few days of meditation, he wakes up one morning and climbs back down the mountain, reaching the bottom a little quicker this time, but nonetheless in the evening. Now, prove that there is a spot that he occupied at the same time of day during the ascent and descent. Good luck. For today's scientific demonstration, we are going to do something you may have seen before on the internet, extracting DNA from cells. That's right, we will be taking out the genetic material that makes living things what they are. Here's what we will need. Organic material to extract the DNA from. We will be using strawberries and chicken liver, but other things such as bananas, blueberries, or kiwis could also be used. Water, dish soap, salt, and isopropyl alcohol. Warning! Be careful when using isopropyl alcohol, as inhaling large amounts of it can cause serious side effects. Before we start, we will want to put our alcohol into the fridge to let it cool down. Now, the first thing that we are going to want to do is take out our organic samples. Here I shall be using 5 stemless strawberries and 2 pieces of chicken liver, one of them which is not shown on screen. To get the maximum amount of surface area, we will crush or grind our product of choice as thin as possible. Remember, while our strawberries or liver are not intact anymore, the cells are still whole for the most part. Next, we shall create a solution by adding 1 cup of water, 1.5 teaspoons of salt, and 3 tablespoons of dish soap. Add more or less 1 tablespoon of the solution per each strawberry that you added, and use a similar proportion for your liver. Mush until the two are thoroughly mixed. Then put each sample through a strainer. Finally, being very careful as to not mix the fluids, pour half a cup of isopropyl alcohol into each sample, and watch as the DNA, including trace amounts of RNA and some proteins, float to the top of the alcohol. As you can see here, this is the part where my liver experiment failed. What likely occurred was that my alcohol mixed in with my liver liquid, which there was quite little of, and likely lacked important components. This in turn was the result of poorly cutting and smushing the liver, which caused problems in the filtering stage. For the future, or if any of you were to try this experiment at home, I suggest grinding the liver in a meat grinder to bring it to a fine consistency first. Out of curiosity, I placed my strawberry sample under a microscope to see if I could see anything of interest. Unfortunately, a lack of dye and lack of strong enough magnification meant that no concrete images were obtained, air bubbles aside. It may seem unexpected that such common household items have the ability to extract the very instructions that make life possible, but considering some of their basic properties, this shouldn't come as much of a surprise. For starters, strawberries were chosen because they are octoploid, meaning that they have 8 complete sets of chromosomes, meaning that they will produce more DNA than many other fruits. Chicken liver was chosen as it is easy to grind and mush up, and serves as a contrast between plant and animal cells. We all know the purpose of dishwasher soap, to get the grease and fat off of plates and other eating utensils. The way soap does this is through emulsification. Lipids, or fats in layman's terms, are hydrophobic. This word comes from Greek, meaning water-fearing, and true to its name, fats do not mix well nor dissolve in water. Soaps, however, have one hydrophilic or water-loving end and one hydrophobic end. When mixed with lipids and water, emulsification occurs. Soap molecules surround the fat molecules with their hydrophobic tails, while keeping the hydrophilic heads on the outside. Such an assembly of molecules is referred to as a micelle, and thus why adding soap while washing hands takes off any grease with ease. This application of dish soap is useful in our experiment, as the exterior cellular membranes, as well as nuclear membranes which contain DNA within, are nearly entirely made of phospholipids. Therefore, the cellular membranes will be partially or entirely destroyed by the dish soap. Next we have the salt. As it should seem logically, all cell parts are soluble or dissolvable in water. They are, after all, floating around in a liquidy cytoplasm. When it comes to salt water, on the other hand, things change a little. Nearly all cellular components are soluble in salt water, but one exception is deoxyribonucleic acid, or DNA, which becomes less hydrophilic due to the ionic environment. At this point, we have an aqueous solution with DNA that is slightly separated and clumped. As a final step, we added cold isopropyl alcohol. While the interaction here is fairly complex, the general idea goes more or less like this. To protect from infections, cells have enzymes whose role is to destroy any DNA that is outside of the nucleus, and lowering the temperature with cold lowers the reaction rate at which this happens. DNA is also not soluble in alcohol, and thus it floats to the top because of its lower density. Meanwhile, other properties of the isopropyl alcohol uncoil the DNA, which is normally wrapped around proteins, and precipitate it, making it appear as the slightly opaque, white, and stringy material that we saw. And that's that. As usual, we hope you enjoyed this week's episode of Science Club. Richard, the editor, and I work hard to make these, so we would really appreciate it if you could like, subscribe, and spread the word about VC Science Club to your classmates. We still have last week's forms up in the description of the video, so it would be great if you could fill that out, especially if you would like to help out with Science Club. Finally, remember to write your answer to today's riddle in the comment section down below for a chance of winning this week's prize, and we'll see you in 2 weeks.